Now we're moving on to fats. Now fats are incredibly important. They're not just there for a fat and energy reserve, but they're also there to make things like hormones and help us make our cell membranes. So they have a ton of roles and a ton of purposes. Now fats, they usually exist as triglycerides, which are just fatty acids and a glycerol. The fat in your diet usually takes that form. The fat that we store usually takes that form. And if you don't get enough fat in your diet, we can actually make some because they're so important. We can actually make them through carbohydrates. Now that sounds kind of funky, but it's true. I mean, if you eat a big carb diet, you know, a big pasta meal over and over and over again, you'll get fat. So if we don't get enough fat in our diet, we can make it through our glycolysis pathway. So again, our nice glycolysis backbone here. And we're gonna now see how fats plug into it. We'll start with citrate, that's part of our TCA. And we wanna go back to our building blocks, in particular acetyl-CoA. And to shuttle citrate back to acetyl-CoA, we need a shuttle called citrate shuttle. So it goes back into acetyl-CoA. And from acetyl-CoA, it gets worked on by acetyl-CoA carboxylase. What do carboxylases do? Move carboxyl groups around with a friend. That friend is biotin, correct? That becomes malonyl-CoA. And malonyl-CoA, through a lot of steps, becomes palmitrate, and eventually we get our triglycerides and our fatty acids and our glycerols. So we don't get enough fat from our diet, we can make it with our glycolysis pathway. Now we can also make something else that is lipid related, and that's cholesterol. Cholesterol you might hear is bad, but things like steroids and hormones and vitamin D all come from cholesterol. So if we don't get enough of that from the dietary fat that we consume, we can make cholesterol. Again, using our nifty building block acetyl-CoA. This time, if you use a different enzyme, thiolase, you can turn it into acetyl-acetyl-CoA. And via HMG-CoA synthase, it becomes HMG-CoA. Now a few more steps. HMG-CoA reductase turns it into mevalonic, and that eventually becomes cholesterol. Clinical importance here, HMG-CoA reductase we can block. If a patient has too much cholesterol in their blood and we don't want it anymore, we can block that and stop the synthesis of cholesterol. The drug that blocks that is your statins. We sure are talking a lot about statins these videos, aren't we? So we just talked about how if you don't get enough fat in your diet, you can make fatty acids, triglycerides, you can make cholesterol. But generally, we do get enough fat in our diets, especially in this day and age. So we don't need to do all of that. So we ingest it, we store it for later use. We have that extra reserve if we need to. If we want to break down fat to give us energy, we can do that in two ways. Both ways require fat to be broken down into acetyl-CoA. And from acetyl-CoA, we can either go into glycolysis, the TCA cycle, ETC, all that stuff. So go into glycolysis, or we can turn acetyl-CoA into an alternative, acetoacetate. And from that, we can give it to our tissues and they can reverse engineer it back into acetyl-CoA, go into glycolysis that way. That acetoacetate is your ketone. When people say ketones, that's what they mean. Basically, acetyl-CoA alternative, acetoacetate. Either way you cut it, the first step is getting it on a shuttle called carnitine shuttle. And that gets worked on by an enzyme while it's on that shuttle called acetyl-CoA dehydrogenase. And the fancy word for all of this is beta oxidation of your fats. Now, before we talk about ketones, I want to go over something that appears on exam from time to time, and that is what happens if you have a deficiency in acetyl-CoA dehydrogenase. Well, you won't be able to convert fat into energy. You won't be able to use it for glycolysis. You won't be able to make ketones from it. So they'll talk about a patient who's starving, who's been fasting or haven't eaten in a while. They'll give you a panel, and in that panel they'll show no ketones. That shouldn't be. If you fasted and you haven't eaten in a while, you should be making ketones. You should be breaking down fat to give you some energy. You should have those ketones. So if you have no ketones, that's a dead giveaway for acetyl-CoA dehydrogenase deficiency. Acetyl-CoA dehydrogenase deficiency. That's it. Don't think twice. It's that simple. No ketones, and someone that should have ketones, someone that hasn't been eating in a while, deficient in that enzyme. Done. So I'm not going to talk about glycolysis. We went over that. I will talk about ketones. How do we turn acetyl-CoA into acetoacetate? Its alternative. Lucky for you, the first half is just like cholesterol synthesis. So, do you remember cholesterol synthesis? First enzyme will be your thiolase. And that turns it to acetyl-CoA. Next enzyme is HMG synthase. HMG-CoA synthase. That makes your HMG-CoA. Now, instead of making cholesterol, we're going to make acetoacetate. And it does that by HMG-CoA lyase. That's the key, that's the difference. And that makes your acetoacetate, your acetyl-CoA alternative. And again, you send that off to your tissue, it can reverse engineer it into acetyl-CoA, go into glycolysis, does what it needs to do. Okay? That is your ketone. Now there's some clinical significance to this. One, if you want to measure ketones, you usually measure this in the urine. Two, this breaks down into beta hydroxybutyrate, and it breaks down into acetone. Acetone is volatile. Hopefully that term kind of triggers something in the back of your mind. You learn about volatility in chemistry. Volatility means it can become a gas. And you can actually breathe out acetone. It gives a distinct smell. It gives that smell, it gives a kind of fruity smell. And people that have a lot of ketones, aka people like DKA patients, diabetic ketoacidosis, they breathe that acetone out. And you can smell it. It smells fruity. And diabetic helper dogs smell that, and they know that their owner is going into DKA. Okay? So that does it for making fats and breaking down fats. Now we're going to see how we transport fats. So how do we move fat around? Fat doesn't do well in fluid, in blood. You can tell that every time you wash dishes, you can see the fat kind of separate from the water, from the fluid. So to get around that, we need to attach it to a protein carrier. And that is your lipoprotein. So lipoprotein is just fat plus a protein carrier, mainly apolipoproteins. And by doing that, we can carry it in the blood and take it where it needs to go. Now there are different categories of lipoproteins, and they're all classified by their weight. Chylomicrons weigh the least, and that's because they have the least amount of proteins. Very low density have a little bit more, LDL have a little bit more, and then HDL has the most. Now HDL has the most protein and the least amount of fat. And because it has that least amount of fat, it can pick fat up. That's why it's called good cholesterol, because it can pick fat and pick cholesterol up. So HDL is good. LDL, on the other hand, has less protein and a lot more fat. High LDL signifies that you have high fat, high cholesterol in your blood. So having too much LDL is bad. That is your lipoproteins. Something to know, these lipoproteins can take in fat via an enzyme called lecithin cholesterol acetyltransferase. And HDL has a special enzyme, takes in fat via this enzyme, and also can pass that fat along, emptying it, and then allowing it to go and pick up more fat. And it does that by transferring fat via cholesterol transfer protein. That's why HDL is so good. Now here's a question. How do we make lipoproteins in the first place? Well, lipoproteins can be made wherever you find a lot of fat and a lot of protein. There are two main areas. One would be in your intestines, and the other would be in your liver. You're going to find a lot of fat and a lot of protein in both these areas, and they can combine together and make lipoproteins. In your intestines, mainly comes in the form of chylomicrons. Those grab a fat, take it to tissue, and if the tissue needs it, it has LPL, or lipoprotein lipase, and that name should give it away. It breaks the fat off the chylomicron, takes it for itself, and what's left is just the chylomicron remnants. And those remnants go to your liver, hit little remnant receptors, get reabsorbed, and then recycled. That's your intestines. Your liver does something similar. Pumps out a lot of lipoproteins, in particular VLDL. Those are low density, very low density. That means a little bit of protein, but a lot of fat. So it pumps those out, goes to tissue that needs fat, and of course those tissue have LPL, lipoprotein lipase. Takes some of that fat away from your VLDL, turns it into IDL, or intermediate density. So that has a little bit less fat, but that keeps chugging along, keeps going to tissue that needs fat, and then another enzyme works on it called hormone lipase. This is just lipase that's more controlled by hormones. That sucks any remaining fat out, and you're left with LDL. And LDL goes back to a nice LDL remnant receptor, gets recycled. There you are. That's how you make lipoproteins, and that's basically the gist of what they do. Taking fat, transporting fat, releasing it to tissue that needs it. I just want to talk about the apolipoproteins for a second here. Certain apolipoproteins have certain functions. So, for example, apolipoprotein A1 activates lecithin cholesterol acetyltransferase. That's the thing that sucks up fat in the first place, keeps it in the lipoprotein. Apolipoprotein B48 mediates chylomicrons. B100 binds LDL. C2 is a cofactor for LPLs. And then E is your remnant uptake. So if you're looking for a state, they'll talk about chylomicrons, and which apolipoprotein it has, talk about LDLs, which proteins it has, HCLs, and they can be difficult to remember, but if you understand the physiology of it, you should be able to reason some of it. So if we take chylos, for example, and we see what they do they pick up fat? Yes. So it must have A1. B48 mediates chylos, so it must have B48. Does it bind to LDL? No, so it doesn't have that. Does it ever get in contact with LPL? Yeah, that's how it releases it. So it has C2. And then finally, is there a remnant uptake of it? Yep. Once it gets used up, the remnants go to the liver. So it has E. So if you understand the physiology, you should be able to reason through a lot of it. Some you might just have to memorize, but I think the more important information is just knowing these in general. The reason for that is if you have a mutation in these, or a mutation in these pathways, you can be in a world of hurt. You can have a lot of cholesterol where you don't want it. These are called familial dyslipidemias. Familial dyslipidemias. The first one is type 1. Chylomicronemia. So you're going to have too much chylomicrons. This is either because of a lack of LPL or the cofactor for LPL, C2. Because you don't have LPL or C2, you can't get the fat out of the chylomicrons. So you're just going to have a ton of chylomicrons sitting around. You're going to have pancreatitis because your blood is just so thick from the chylomicrons. Hepatosplenomegaly because of this engorge. And if you spin the blood in a centrifuge, you're going to have a top layer of fat called creamy supernatant. So those are the symptoms of type 1. Type 2 is hypercholesterol. This is a defect in your LDL receptors or the protein for it. And because of this, you can't take up the remnants of LDL, recycle it. You just have LDL floating in your blood. This is the most dangerous. You're going to have so much fat you can see it in your eyes. You'll have a ring of fat called corneal arcus. And almost pathognomonic, you're going to have a lump of fat in your Achilles tendon called tendon xanthoma. Tendon xanthoma. But the thing that kills you are going to be myocardial infarctions. You're going to have very advanced atherosclerosis disease. Why is that? Well, in atherosclerosis, when you have vessel damage, fat can go in there, macrophages eat it up, become foam cells, release factors, causes the whole atherosclerosis. I won't get into it. But the fat that gets in there initially, is that not LDL? Yes or no? It is. That's why if you have too much LDL, you can have MIs. In type 2, you have too much LDL, and that's why you have those MIs. So type 2 is deficiency in LDL receptors or ApoB100, the most deadly, okay? Lastly, type 4, hypertriglycerides. This is either too much VLDL, you're pumping it too much, and your body breaks it down, takes those fat from your VLDLs, but because there's too much, it's taking in too much fat. You have hypertriglycerides. And the main symptom is just pancreatitis from having too much of it in your blood. Your blood gets thickened, your pancreas gets clogged, you get pancreatitis. Hope that makes sense. Know these, know familiar dyslipidemias, and then kind of just broadly know the physiology of it. Never seen a question about the physiology of it, but I have seen questions about what goes wrong. That does it for this video. See you next time.